Hello and welcome back to the Soil Health and Nutrient Management section of our Master Gardeners class. I'm Jason Clark and I'll be your instructor again for this video course that we're on right now. And again, as we've been talking about previously, we're working on what we need to do to improve our soil properties with that goal of maximizing the growth potential of our plants. And we've been focusing on how we can improve the 6 different areas that we can see here. And up to this point, we've covered the first 5, from nutrient retention down to pH. In this section, we'll be covering number 6, availability of nutrients. This is actually one of my favorite sections because it's a lot of the work that I do in my job is figuring out how to make the nutrients available that our plants need to obtain that optimal growth. So that's what we'll get into in this video here. So first of all, we've already talked about the 14 essential nutrients that come from the soil, and those nutrients are spread out in the soil. And what that plant root does is it goes down to the soil and it can take up some of those nutrients. Some of those nutrients it needs it right by the root in order to take them up, like we can see here, and utilize those nutrients. Some of them can move through the water as what's taking up water, move a little bit easier, such as nitrogen and sulfur. However, as the plant gets older, it still sends its roots further out into the soil and it continues to take up the nutrients that it needs. Nutrients can move some in the soil. Some nutrients move slowly, such as phosphorus and potassium. Nutrients such as nitrogen and sulfur move a little bit more readily in the soil. So we can use these nutrients the plant needs to really grow, to stay green, to stay healthy, produce the fruit, or whatever we're going to eat of that plant. And there are several different ways we can determine if we have sufficient nutrients in our soils for our plants. And the main way we can do this is by soil testing. So we take a sample of soil, we test it to determine how many nutrients are there and if we have enough to support the type of plant that we're growing. Another thing we can do is plant tissue testing. In this, we take tissue from healthy plants, tissues from plants that seem to have a problem or aren't growing well. We analyze them for nutrient content and we compare which nutrients are missing or they're not as abundantly in the non-healthy area as in the healthy area. The last thing we can do is we can look at deficiency symptoms, and this is usually a yellowing of different parts of the plants. So first of all, we're going to touch on soil testing and how soil testing can help us improve the production of our plants, our gardens, our flowers, trees, and shrubs. So the first thing to do is, we've kind of talked about this a little bit, but soil testing labs determine several different factors. And one of them is soil physical characteristics. So they determine texture, the amount of sand, silt, and clay. So then we can use that information to determine if it's a loam, a clay loam, a sandy soil, of that nature. And we can use the mechanical sieve analysis or also that hydrometer method that we talked about in an earlier video. Another thing we can do besides soil physical characteristics such as texture, we can look at those chemical measurements. Those chemical measurements include pH, which pH is important because it determines the availability of certain nutrients, and also organic matter, because that black gold that we want abundantly in our soil that can help with filtration, it can help with drainage, help with keeping our nutrients in the soil instead of losing them. Another thing we can look at is salt affected soils. Do we have too many salts in our soil? So we get certain levels of salt that decreases the growth of our plants. And we can do this in several different ways. There's things that measure electrical conductivity, such as EC, that you may hear about. They also use another couple of procedures called saturated paste or extractable paste. Most likely you'd be using and asking them to look at electrical conductivity. The next thing they're going to do besides soil physical or chemical measurements, and we can have them do one of these, all of these, or just a few of them from each category, is they're going to look at the status of the amount of nutrients that are in our soil. Specifically, they're going to look at nitrogen, phosphorus, potassium, sulfur, calcium, boron, any other micronutrients that we may ask them to do. And then after they have given us that, they'll usually give us some sort of recommendation, because we're usually going to tell them we're growing flowers, we're growing shrubs, we're growing grass, we're growing vegetable garden. And based on what we know will be going into that area, we can know how much of which nutrients we need to be in that soil. So sometimes we're sufficient, and other times we need to add certain nutrients. So timing of soil testing is important. We can test in the fall or the spring, but the important part is to do it before we add compost, manure, or fertilizers. so we know what is already in the soil for the next growing season or the upcoming growing season. And we can sample every year, or we can sample every 2 to 3 years, and that really depends on the soil tests we're doing and the nutrients. Some nutrients move around a lot in the soil, or else they're broken down from organic matter, so we need to test them every year. Other nutrients are fairly stable. So if we want to test for nitrogen, we should do that every year, because nitrogen is very, what they call biologically mediated or microbially mediated. So microbes are very much the dependent factor on how much and when that nitrogen becomes available for the plant. And because it changes annually, we test it annually. Whereas if we contrast that to tests such as phosphorus and potassium and micronutrients such as zinc, manganese, boron, molybdenum, etc., we can test these each year, but most likely they don't change a whole lot from year to year. So we test them every 2 to 3 years. Because these are more stable in the soil particles hold on to them. There's just the mineral component of the soil that we mentioned in our first video. A lot of these nutrients come from that mineral content of the soil. The next question, so after we know how often we're going to sample and when, we need to know where to sample. So there's a few steps here, and we'll give an example. First thing we need to do is we need to divide the sampling areas by use. So divide these areas that we're going to soil sample into a lawn area, into a garden area, into a flower bed area, into a wooded tree area, and then we're going to sample these areas separately. And when we're sampling, a couple of important things to remind ourselves of and avoid is we want to sample separately or avoid sampling certain areas. These are low spots, areas near trees or buildings and borders. For example, if you went to the lawn at my house, you would see a big pine tree out in the front yard, and underneath that pine tree you'd see, during times of drought or a little bit less water, you see this greener ring around that pine tree because there's a little bit more organic matter underneath the pine tree, and that soil is different. So outside of that area it has a little bit of a trouble growing sometimes, needs some more help, but underneath that tree it's a darker green color. So if I were to sample both areas together, we might not truly know what the rest of the lawn needs because they're different from each other. Another thing we need to do is we need to randomly sample each area. Don't just sample a little 5-foot square area, but randomly choose different areas in there to get, and we'll look at a diagram here really quickly of a lawn. So if we look right here, we can see a few different things. We can see our house right here, and then we have our green grass area is one area that we can sample. Then over here we have this low-lying area, maybe shrubs or trees, a little bit farther away from the house, a little bit different. A wooded area, and then back here could be maybe our vegetable garden. We need to sample all these areas separately. And sometimes even within our vegetable areas, maybe this area over here we're having trouble. We seem to not be able to grow our tomatoes or our peppers very well, but over here we don't have as much of a problem. So maybe I sample this area with one soil sample and this area with another soil sample, and then compare them to see maybe what nutrient might be missing over here. But this is kind of a diagram of how we'd want to separately sample, our yard or different areas like that. The next thing we want to know is after know where to sample, we need to know the depth that we need to sample. And the depth depends on the plants grown and how deep their root systems are. Because if we sample down a foot or two, but our roots don't get down that far, we're going to get not an accurate estimate of how many nutrients are available for our plants. So for existing grass, we only need to sample the top 3 inches. For a new grass seeding, we want to sample the top 6 inches. For looking at gardens, about the top 6 inches as well. And trees and shrubs access nutrients a little deeper, so we're going to go from 0 to 12 inches. Alright, now the next step is we need to learn how to take an appropriate sample. So with this, we need some tools. So we're going to get the garden trowel, the shovel, the spade, or we can use the sampling tube, depending on which of these 3 items that we want to use. A lot of times I use sampling tubes because I sample a lot, but you can find these in some of your garden stores or hardware stores to be able to get those. And we want to scrape away the surface mat or the litter. So you can see here the soil sample was taken, but then they took off that grass mat layer on top. Now you can do this before you take the soil sample, or you can do it after. Either way is fine, but it's important to take it off because we don't want that organic matter in our soil sample, because it'll change the analysis that we might get back from the lab. The other thing we want to do is we want to sample that garden or lawn area to the depth indicated, whether it's 3 inches, 6 inches, or 12 inches. And this is just a couple diagrams depending on the equipment that you have to use. So for a soil tube, you just go straight down 6 inches in the soil, pull it back up, take that soil tube out. If you're using a garden trowel or spade, you dig a little bit of a hole right here, move the soil to the side, and then you take about 1 inch farther over and you dig down. And you're going to take about a 1 inch by 1 inch square, and that's going to be your soil sample. So you're going to cut off the edges of the sample, so you just have that middle square inch rectangular piece that you're going to put into your sampling bucket, which is next. So we put that into our sampling bucket, and then we're going to do this process again in about 10 different spots randomly throughout the lawn, throughout the garden, the area that we're sampling. The next thing to do is to mix that sample really well inside that bucket. Break up the clods, break up everything, mix it really well. And then from there, we're going to take that soil sample, and we're going to take about 2 cups. And the 2 cups is what we're going to end up sending to the lab. But before we send it to the lab, we want to put that sample, spread it out, let it dry overnight, put a fan on it to help it dry out. And we're going to send that to the lab in order to get our analysis. And for specific labs, they might have a little different procedure. Maybe they want it in a plastic bag or paper bag. Maybe they want a form enclosed with it. So it's best to get on their website, give them a call. If you want to go on the extension website, there will be a list of labs that are available that we can send our soils to. The cost is fairly minimal for gardens or things like that. I think it's 15 to 20 bucks a sample, depending on the lab. So from there, the next thing we want to do, if we want to look at nutrient status of our plants, is we can look at the deficiency symptoms that we mentioned earlier. So plants, when they're deficient in certain nutrients, they can show that symptoms. And so the goal of this is I want you to be able to, after we're done with this next section, look at these 3 corn plants, these 3 pictures here. So 1, 2, 3. And I want you to be able to look at the different symptoms you may see here and be able to identify what is deficient in these 3 different corn plants. So let's get started on kind of learning about what they are. But first of all, we'll just do a little bit of general information. So when we observe nutrient deficiency symptoms, commonly it's going to be a yellowing of a specific plant part, and there's going to be a pattern. So if we look at this plant right here, we can see different yellowing patterns. We can look over here, we can see some yellowing there, some yellowing here, some yellowing over here. And based on where that yellowing is and to what degree, it is most likely going to indicate a deficiency of some sort. However, the caveat here is these aren't always definitively a deficiency. Sometimes there's a weather issue or other things like that. But we do have certain types of situations where it's fairly common to see deficiency symptoms in plants. So these commonly occur in new garden areas. So if we haven't planted a garden there before, the soil has been used for something else, or it's been bare for a long time, sometimes we're missing the microbes, the fungus, and other types of living organisms that are needed to help get those nutrients to the plants when they need them. They might be in the soil, but they have a hard time getting them to the plants in that first year. It's that same idea if we're growing a new vegetable or fruit crop for the first time. We might see some deficiency symptoms where in the next years we won't. And then again, as we mentioned earlier with phosphorus, we can sometimes see certain deficiencies such as phosphorus due to unusual weather patterns. Maybe we go hot, dry, wet, dry. Or else it's really cool in the spring, so microbes can't break down certain nutrients to make them available to crops. All these different things can affect availability of nutrients. And sometimes we can even see deficiency symptoms if we apply too much of certain nutrients. So if we over-apply limestone, if we over-apply phosphorus, or add too many micronutrients, we can sometimes get toxicity symptoms, which look similar in some cases to deficiency symptoms. And then of course, if we haven't managed our soil very well in the past, we could see some issues arising from that as well. The first nutrient deficiency we'll talk about is nitrogen deficiency. Nitrogen deficiency is shown by a uniform, gradual yellowing of lower leaves. The lower leaf portion is very important because the upper leaves, if they start to get yellow, that's a different nutrient deficiency. That might be sulfur. However, the lower leaves is important because nitrogen is mobile. It moves around in the plant quite easily. So as a plant is growing, if it's starting to produce leaves at the top of the plant, there's not quite enough nitrogen. What the plant will do is it'll move that nitrogen from the lower leaves into the upper leaves, and when it moves it from the lower leaves to the upper leaves, it leaves those nutrient deficiency symptoms of yellowing of the lower leaves. So there can be several causes. One can be we just didn't apply enough nitrogen from our compost with our fertilizers so the plant could have enough when it needed it. Another thing could be we could have applied enough nitrogen at the beginning of the season for the plant, however, we had a lot of that nitrogen move down and out of the root zone due to leaching. And that nitrogen moves out of the root zone due to excessive precipitation or irrigation. So the nitrate is negatively charged, which is the most common form of nitrogen in the soil. And because it's negatively charged and the soil particles are negatively charged, they repel each other, so the soil particles don't hold on to the nitrate form of nitrogen as well as it does other nutrients, so it moves down and out of that root zone due to too much precipitation or irrigation. So we could have applied enough, however, because of precipitation and irrigation events, we move that nitrogen down and out of the soil. So a lot of times they'll say to split up that nitrogen application, apply it once every 2 weeks, once a week, just in small amounts as the plants start to grow more. The next thing, not only can we lose it because we have too much water, it moves it down. If we have inadequate drainage, that water just sitting on the top creates saturated environments. All that water displaces the oxygen, and then what happens is those microbes that like conditions where we don't have nitrogen or we don't have oxygen, anaerobic conditions, they'll convert that nitrogen into a gas and release it into the atmosphere. And so we can lose nitrogen going down too far in the soil profile and also volatilizing it up to the atmosphere. And also, as we mentioned earlier, some plants purposely will lose their lower leaves at later stages of growth. Maybe because they don't need those lower leaves as much, because we have upper leaves that are kind of shading out the lower leaves. Just kind of depends. But the correction for nitrogen deficiency is to apply fertilizer, compost tea, a form of nitrogen that's readily available for the plants. This can be done foliarly, depending on the product that you use. Nitrogen deficiency is something, if you catch it early, you can still get it fixed and get enough nitrogen there, because it easily moves with the soil water and into the plants through irrigation and things like that. So if we want to look at some symptoms here, we can look at some corn symptomology here. Again, we see the yellowing of these lower leaves. The green leaves up here, the upper leaves are still green, but these lower leaves start to turn yellow because that nitrogen, the plant is moving from the lower leaves to the upper leaves. And here's just kind of a cool picture showing nitrogen at different rates. So we have a sufficient rate of nitrogen up here, so we see how big and tall this leaf is and wide. And as we reduce the amount of nitrogen we apply to the corn crop, we can see the leaves get a little bit smaller. And then they start to yellow here, just some yellowing, and then this whole leaf is almost all yellow. We can also see the yellowing of the lower leaves of a cauliflower plant. Then here, this is the lower leaves of a tomato plant, and they're starting to just get this general yellow form, and that's nitrogen deficiency symptom. So if we switch gears now to phosphorus deficiency, we see phosphorus deficiency is a little bit different. It's not a yellowing, but it's actually a purpling of leaves and short plants. And the cause to phosphorus deficiency is commonly wet and cold soils in the early part of the growing season. And a lot of times plants will grow out of this. Once the soil warms up, microbes start doing their thing, decomposing organic matter, taking that phosphorus that's in the soil, just not available, making it available to the plants. Another thing, of course, as always, if we don't apply enough nitrogen for the plant to grow well through compost or fertilizer, then we're going to see deficiency symptoms of phosphorus. And this is more sensitive in coarse textured soils than fine textured soils. So in order to correct this, what we'd want to do is we want to apply a subsurface fertilizer, a compost tea, or something similar to that nature. And these a lot of times are called rescue treatments. We're hoping for the best, hoping they work, and they can work if they're caught early enough, but they're not as easily treated as something such as nitrogen. But it's important to get this below the surface of the soil or in a form that the leaves can take up, because phosphorus doesn't move well in the soil, it kind of stays put. And so the roots have to grow out to it and pick up the fertilizer. So if we add it, we need to add it below the soil surface, somewhere where the roots can get at it quickly. If we look here at a couple pictures of phosphorus deficiency, again we can see the corn here, and we can see the purpling of the stem purpling of these leaves. And if we compare this against a corn plant without phosphorus deficiency, the one without phosphorus deficiency is probably going to be a little taller. Now here, if we contrast this and we look at some tomato plants, we can see two different groups here. A phosphorus deficiency group over here on our left, and a normal group over here. We can see the normal group, dark green leaves, taller, phosphorus deficiency group, purpling of the stem, purpling of the leaves, stunted, shorter plants. Now moving on to potassium deficiency. This one is again a yellowing, but it's a yellowing of leaf margins. The margins of the leaf are those edges of the leaf. So the things that cause this are going to be insufficient potassium that we add to our soil to support the plants, either from compost, fertilizer, or some other source of nutrients. And again, just like phosphorus, coarse textured soils tend to be more sensitive to having potassium deficiency. And you're going to correct this in a similar way that you're going to correct phosphorus. You just need to have subsurface supply, some sort of fertilizer, compost tea, etc. And again, these are more rescue treatments. You're just hoping maybe you can do it, and that's if you catch it early enough. So if we look at the deficiency symptoms of potassium, we can see here that we have yellowing, but it's not the whole leaf. This middle part is still green. It's just these leaf edges. So this is a picture of some corn in a trial that we're looking at some potassium fertilizers. So we can see the deficiency symptom there. We can also look at it in ornamental plants. Just that leaf edge will start to get yellow and brown. Tomatoes, again, that leaf edge, we start to see this area here get yellow and brown. Cabbage, again, that outside leaf margin, not the whole leaf, just that outside portion. Peas, we can see it here. Raspberries, again, that outside margin of the leaf. So now moving on to calcium deficiency. Calcium deficiency is a little different. We're not usually going to see this for the plant. We're going to see this for the fruiting body of the plant. It looks kind of like a rottenness of the outside or the inside of these fruiting bodies. And we'll see some pictures in just a second. So some causes of calcium deficiency are highly acidic soil pH. We have really acidic soils, calcium's not available, it's usually not present in good amounts in that soil, so we need to add some calcium to that soil that's highly acidic. Another one is if we have a lot of wet and dry cycles, so if we have moisture stressed environment and then we get a huge rainfall or a big irrigation event, we can start to see these calcium deficiency symptoms as well. And the best way to correct this is really to add calcium maybe in preseason to up that pH, as we see here, and also to irrigate or kind of lessen the time between maybe moisture stress and wetting cycle. So maybe we irrigate if we haven't gotten precipitation in a while. So here we have our watermelon plant, and we can see this kind of rottingness or that browning of the fruiting body. Again, here with apples, we see these spots here. You have to be kind of careful because some of these can be actually due to insects and not calcium deficiency. Tomato plants, you see this rottenness of the bottom layer of the fruiting body. Moving on to iron deficiency. With iron deficiency, it's a little different. We're actually seeing a gradual yellowing of upper leaves between the veins. So the veins themselves, we have this dark green vein, but the area between the veins is a yellow. It's called interveinal chlorosis. And this can be caused by alkaline soil, so our soil pH is above 7.5. And if we remember back to our pH lesson, iron is available at lower pHs. Once we start to get above 7.5, 7, our iron becomes less available. So if we have alkaline soils above that, it can likely cause that iron deficiency. If we're irrigating with alkaline water, and that's kind of increasing the pH around our plant roots, they're taking up that water, that's going to make iron less available. And then also if we're over applying limestone or wood ash, which raises our pH, as we learned about earlier in another video, we could be lowering the availability of that iron nutrient. So iron availability and that deficiency symptom is a lot of times tied to pH of the soil. So for correcting iron deficiency, there's usually a lot of products out there that have some sort of iron fertilizer that we can add, and we can apply that foliarly to the leaves. And if we want to look at this more long term, we want to try and lower the soil pH over time by adding sulfur or some other pH lowering product. If we want to look at some symptomology here, we can look at our strawberry plant. We can see here the veins still have this dark green color, but in between the veins they're quite yellow. Same thing with our grape. The vein is dark green, but in between those veins you can see the yellowing color, as we can see here and throughout. Similar concept with our blueberries. Maple trees have the same effect. Oak trees, green ash, and sometimes it only affects part of the plant because certain parts of the root system take up the iron and take it to the plant, so the pH of the certain areas around our tree can also cause this. Apple trees as well. You can see that yellowing between the veins, but the veins are still a dark green. So that goes on to show some of the nutrient deficiency symptoms that we're commonly going to see in South Dakota. There are other nutrient deficiency symptoms we might see. For example, sulfur we might be able to start to see, especially towards the eastern part of the state or in soils that have low organic matter content. And for those plant tissue deficiency symptoms, we're going to be looking at kind of a yellowing or a striping of the top portion of the plant. Remember, nitrogen is that yellowing at the bottom of the plant. Sulfur is kind of yellowing or striping at the top of the top leaves. So also we may want to analyze our plant tissue to see what nutrients may be missing or not in sufficient amounts. A lot of the times tissue analysis is only really cost effective in high scale commercial production or fruit production, but it can also be used if you just have a persistent problem in your yard, in your garden, your trees, and you're just not sure. Some areas you don't see it, other areas you do. You can use plant tissue analysis in this case to figure out what might be the issue or the problem. So if we want to do a plant tissue analysis, how do we do it? This is a really important problem. So the first thing we're going to do is we're going to sample the problem area, but we're also going to sample the healthy area. So here for this example in this field, we see a problem area. Here we see this stunted plants, yellowing of the leaf tissue, but over here we also see a healthy dark green tall plants. So what we'll want to do is we'll want to take samples of the leaves and or the stems, depending on where that deficiency symptom is showing up. Sometimes you can take the whole plant if it's throughout the whole plant. If not, you just want to take the leaves that are showing the symptom and then the healthy leaves. And then from there you're going to have those two samples, your healthy sample, your problem sample. You're going to dry those samples and you're going to submit them to the lab. And if you check on the lab websites, they're going to tell you a little bit more about how to do this, how to send in that sample, how to dry it, things of that nature, how much leaf material they need. But what you're going to do is, since you have a health area and a problem area, the lab is going to analyze that plant tissue for nutrient concentration and how many nutrients are in those leaves, in those leaf tissues, and you're going to compare the healthy leaf tissue to the problem leaf tissue. And then from there what we can do is see which ones are missing in the problem tissue. Maybe we have a lot lower potassium or phosphorus or sulfur, and the healthy tissue has a lot more of those, so we can kind of know, okay, it's a sulfur issue. We need to most likely maybe apply sulfur to this area, or potassium or phosphorus. So when we use plant tissue nutrient analysis, or we're seeing a deficiency problem in our plants, some of them we can correct quickly, some of them we're probably going to have to wait until next year to correct. So things like nitrogen deficiency, if we catch it easily, we can immediately kind of take care of those. We can add some nitrogen fertilizer, we can add it foliarly if we need to, if we're careful, or a compost tea of some nature. We're looking at some micronutrients. A lot of times there's micronutrient foliar applications that we can do, or we can apply them in some other way, and that can be immediately done through a foliar micronutrient. And we want to make sure it's certain micronutrients that we're foliar applying, so we're using a chelated version. What that chelated does is it's something that holds on to that nutrients. So for example, iron. Iron, if we put it on soils, high pH soils, it's going to react very quickly with calcium and not be available for the plant. However, if we have a chelated form, that chelate holds on to the iron, and then once calcium or whatever shows up to grab the iron, the chelate says, nope, I've got this, you can't touch it, and the plant can still take it up. So for a lot of other nutrient deficiency symptoms, we see them, we can try to correct them. However, a lot of times what we're going to have to do is just better fertilize or add more compost to our soil the next year. Alright, so we've successfully navigated our way through how to improve the availability of our nutrients, how we can take a soil test so we can know what nutrients are available and in what quantity, how we can recognize plant tissue deficiency symptoms, and how we can take a soil and a plant tissue test so we can kind of compare those and know what nutrients might be missing. As always, thank you for your time, and of course, reach out to me for any questions you may have. Thank you.